Dear friends, today we are going to study about composition, structure and function of carbohydrates, part 8. In this part, we'll study about glycoconjugates. Before that, we should know what is the function of carbohydrates. So the basic function of carbohydrates we already studied in the last videos. That is, they act as a stored fuels, they act as a structural elements, and they also function as an extracellular support. Apart from these basic functions of carbohydrates, carbohydrates are also used in various other functions like in cell-cell recognition, cell adhesion, cell migration, blood clotting, immune response and wound healing. For all these functions, the carbohydrate must bind either with a protein or with a lipid. And that binding is known as glycoconjugates. So what are glycoconjugates? It is a conjugate between carbohydrate and protein or lipid, and this binding involves covalent linkage. So carbohydrate covalently bind to protein or lipid to form glycoconjugates to perform these additional functions of cell-cell recognition, cell adhesion, migration, blood clotting, immune response and wound healing. Now, what are proteoglycans? Proteoglycans, these are macromolecules found in cell surface or extracellular matrix. In proteoglycans, it is a combination of glycans. Glycans means glycosaminoglycans. In short, it is known as glycans. So, proteoglycans is a combination of glycosaminoglycans and protein, which are joined covalently. In this combination, the glycosaminoglycan, they constitute the main part of the conjugate. This glycosaminoglycans only function as the main part of biological activity. It is bind with a protein. Proteoglycans form the major component of connective tissues like cartilage. It interacts with other proteoglycans and proteins with non-covalent interaction to provide strength. What are glycoproteins then? Glycoproteins is combination of protein with oligosaccharides. Don't get confused between proteoglycans and glycoproteins. Proteoglycans is also a combination of carbohydrate and protein. Glycoproteins are also a combination of carbohydrate and protein. But in case of proteoglycans, the protein is paired with glycosaminoglycans, whereas in glycoprotein, the protein is paired with oligosaccharides. So glycoproteins are the conjugate in which several oligosaccharides join covalently to a protein. Where we can find glycoproteins? On the outer side of the plasma membrane, in the extracellular matrix, and as well as in the blood. Inside the cell, we can find glycoproteins in Golgi complexes, secretory granules and lysosomes. They act as a specific site for recognitions and protein binding. What are glycolipids? Where carbohydrate is attached with lipids, and where we can find lipids? In the membrane. So in glycolipids, membrane lipids containing hydrophilic head groups are oligosaccharides. Next is proteoglycans are macromolecules of cell surface and extracellular matrix. Mammalian cell contain at least 30 types of molecules of proteoglycans. So these proteoglycan superfamily, they function as a tissue organizer for the development of specialized tissues, to mediate various growth factors, and to regulate extracellular assembly of collagen fibers. A proteoglycan unit consists of a core protein and attached to it is a glycosaminoglycans. Now this is the basic structure of a proteoglycan. What we can see in this structure? This serine, glycine, X serine, that means this is a core protein. As because it is a protein, it will have an amino terminus and a carboxyl terminus. Now to this core protein, glycosaminoglycan is attached. The attachment point is serine in the protein, to which the glycosaminoglycans are attached. Now this is an example of a syndecan core protein. What we can see here? It is a membrane protein which consists of a single transmembrane domain and an extracellular domain. Now in this extracellular domain, there are 3 chains of heparin sulfate. The green chain is a heparin sulfate. So how many green chains? 1, 2 and 3. And 2 chains of chondroitin sulfate, the orange one. So this is a typical example of a syndecan core protein which consists of a single transmembrane domain and an extracellular domain. In mammals, we have 4 members of syndecan family which interact with growth factors, adhesion receptors, cytokines, chemokines, proteinases, and other extracellular matrix proteins and alter or help to perform their function. Now one is heparin sulfate moieties, which are found to be attached with many core proteins. Now in heparin sulfate moiety, it is a disaccharide repeating units. Heparin is a disaccharide repeating unit in which the monosaccharides are glucuronic acid and N-acetyl glucosamine. These units are repeating units in case of heparin. First, these are repeated 50 to 200 times. After the repetition, the next step is it is acted upon by various enzymes, among which the first enzyme is N-deacetylase N-sulfotransferase. What could be the function of this enzyme? It removes acetyl group and in place of acetyl group, it will transfer sulfate. After this, the next enzyme will come into play and that enzyme is epimerase. It will convert glucuronic acid into hydronic acid. Then the next enzyme will come, which is sulfotransferase. It will add sulfur group at C2 position of hydronic acid and C6 position of N-sulfated glucosamine. The addition of sulfur won't happen at all the places. This disaccharide is repeated 50 to 200 times. Now, if it is repeated 200 times, then in all the 200 places, sulfur group won't be added. So, for different protein, the place of addition of sulfur group is different in case of different proteins. As well as it is also different in case of same protein found in different cells. Heparin sulfate moiety, it binds to a variety of extracellular ligands. The glycan moiety of proteoglycan has revealed a sequence heterogeneity that is not random. That means there is a difference in sequence homogeneity. As I said, it is repeated 50 to 200 times. With some protein, it may be 50 times. With some protein, it may be 100 times. So it will differ in length. It will also differ in sequence where the sulfate group has been added. So the exact pattern of sulfation in the S domain. If you see this structure of heparin moiety, two domains are there, S domain and Na domain. As I said, all the place won't get sulfated. Some place will get sulfated, some place won't get sulfated. So where sulfur is added, that is known as S domain, rich in sulfur or rich in sulfate. Na domain means where sulfate group has not been added, it is the original one, that is known as Na domain. So the exact pattern of sulfation in the S domain that differs in different proteoglycans as well as it differ in same proteoglycan if it occur in different cells. Proteoglycan number 1, proteoglycan number 2. Addition of sulfate will be different in both the proteoglycans and also in the same proteoglycan if it occur in cell X and it is occurring cell Y. It will be different in different proteoglycans. It will be also different in same proteoglycans if it occurs in different cell types. Next, if we see the S domain, it binds specifically to the extracellular proteins and it acts as a signaling molecule to alter the activities. It helps in the activity of other proteins. It helps in cell-cell interaction. It helps in cell signaling. But there are 4 different ways by which it can do. It can perform its work. What are those? First is conformational activation. What does this mean? You see the green one is the S domain. So what is happening? AT is antithrombin. So first the antithrombin will bind with the S domain, due to which the conformation of the antithrombin will change in such a way that it can bind with the factor XA. And hence the process can start, the process of blood coagulation. So this is one way by which the S domain alters the activity of the extracellular factor. Next mechanism is enhanced protein-protein interaction. What is happening here? The antithrombin is bind with S domain, due to which what is happening? To bind antithrombin and thrombin, first the antithrombin bind with the S domain and the thrombin will also bind with the other S domain. Due to this binding, these thrombin and antithrombin can bind with each other. So this is known as enhanced protein-protein interaction. Antithrombin is also a protein. Thrombin is also a protein. These two proteins can interact only because of S domain of heparin sulfate. Because these two can bind with the S domain, Therefore, they can able to bind with each other. Next mechanism is co-receptor for extracellular ligand. What is happening here? In the extracellular region, the ligand is present. What is the ligand? FGF. FGF means fibroblast growth factor. So growth factor means it will help in the growth. It will help in cell division. Because of less concentration of this growth factor, it cannot bind with the receptor efficiently. So what is happening here? Because of S domain, the local concentration of ligand will increase, which will bind with the S domain of heparin sulfate of syndican molecule of target cell. Cell growth. The function of FGF is cell division. So if this particular cell needs to divide, then in this cell membrane, the syndican molecule will be present. In that syndican molecule, the heparin sulfate will be present. In this heparin sulfate, the S domain is found. In this S domain, the fibroblast growth factor will come and bind, and hence it can interact easily with the receptor. Then the cell signaling or signal transduction process will start and helps the cell to divide. The next mechanism is cell surface localization or concentration. It is also the same type. In the S domain, here the example is of lipoprotein lipase. The lipoprotein lipase will bind because S domain contains sulfate, so that means it is negatively charged. Lipoprotein lipase is a positively charged molecule. So electrostatic interaction will come into play, due to which the lipoprotein lipase will come and bind with the S domain. Such type of interaction we can also find during the infection of the virus into the cell. So these are the 4 ways by which the heparin sulfate, or specifically the S domain of the heparin sulfate, can alter activity of other proteins or molecules so that they can function efficiently. Now one term is proteoglycan aggregate. Aggregation means accumulation of many proteoglycans. So when many proteoglycans accumulate, they form an enormous supramolecule assembly having many core proteins, and they all bound to a single molecule of hyaluronate. So this aggregate core protein, they contain multiple chains of chondroitin sulfate and keratin sulfate. How they joined with the core protein? Through serine residue. They interact strongly with collagen in extracellular matrix of cartilage, and hence they provide strength to the cartilage. Due to this proteoglycan aggregates, what happens is the one proteoglycan is joined or cross-linked with other proteoglycans and also with other proteins like elastin, fibronectin, and because of this cross-linked meshwork, the extracellular matrix gain its strength. If you see the diagram, you can see here, this is a hyaluronate. This center one, this center line is of hyaluronate. With this center one, keratin sulfate, and this is a center of hyaluronate. Like this, keratin sulfates are attached, and with this, chondroitin sulfates are attached. So this form the aggregate core protein. So this aggregate core protein, because it is an aggregation of many proteoglycans, also proteoglycans with other proteins like elastin, fibronectin, they form a cross-link meshwork which gives the extracellular matrix high strength. Now one question from my end for you all, that is, what will happen if S domain is knocked out? So you have to answer what will happen if S domain is missing. We already studied various functions or the importance of S domain. So you have to tell me what will happen if any cell membrane or if in a cell the S domain is missing from the heparin sulfate. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.